try to make it worth your while. Well, I can start by making, well, no, I'll hold that for just, let me start by making just clarification of what's available to you on Moodle. So here are the lists of open houses that we've already had. Today, there's more time for cadaver review. This is cadaver open house. Cadaver review can happen today and tomorrow. But then they stop. Those of you who are in the Thursday evening lab will do the practical quiz this evening. Those of you who are in the Tuesday lab have some options. Here are the practical quiz options for you the regular time, Tuesday afternoon, or some other options here. I'm not asking you to tell me what you want. I'm assuming that you know that you have to come to one of these options. And the proof that you attended will be you turn in your answer sheet. That's all. There's going to be no sign up for that. So this will be somewhat fluid. What I'll do is I'll wait until a half-hour mark, either on the hour or on the half hour, unless there's a group of 4 students who are ready to go, in which case we'll just start. So we'll start as needed. And if there's only 1 student, we'll start, or 2, we'll start at that half hour or the hour. But if there's 4 students, we'll start whenever there's 4 students. Sort of the same thing with the exam, right? If you're going to choose one of these, here's the regular exam time, 8:30 in the morning here on Tuesday. But if you're going to choose one of the other options for the exam, you just have to get the exam from me. And if I'm not in my office, you'll get the exam from the biology department office, and we'll set you up with a place to take the exam. Any questions about this somewhat fluid set of requirements? It's fluid in the sense that there's lots of options. It's a requirement in the sense that you have to take the quiz and the exam. Okay? So today is a question and answer session. As usual, I have some slides that we can use as the basis for a conversation, but really what I want to do is answer your questions, making it worth your while getting up this early on a Thursday morning. But to make it worth your while, here is a life-size model of the human clitoris. So let's get oriented. This is anterior, posterior, lateral, lateral. Right? So this is the front, this is the back. This is life size. So this is what women have inside their bodies. Only about that much of it is visible. So the rest of this structure is hidden inside the labia. Not hidden, I mean that's where it is. It's inside the labia. So the urethra would be, right about here. So this is now front. Urethra and the vaginal opening would be around here. Okay, so front. Well, I should say the opening would be about here because this is deep in the labia. This part. These sort of fine, narrower structures are the corpus cavernosum, and the broader structures are called the bulb, which is also part of the corpus cavernosum. So this entire structure, both of these, are highly vascularized tissue that engorge with blood during sexual arousal, just like in men. I'm going to pass this around. I got this from Maggie Thomas, who teaches women in gender studies as well as psychology. So before so here's a slide just to have on the screen, right? In the hypothalamus, the green things are neurons, the red things are microarteries, the blue things are the microvasculature of the median eminence, where these hormones are released. And then here's a cartoon of the anterior pituitary, the vasculature, and the posterior pituitary with its nerve terminals from the hypothalamus. We don't have to talk about this, it's just that's where we sort of started the endocrine section. Yes, Shannon.
I'm actually confused about this a little bit. So for in terms of the anterior part,
Yeah.
so it goes, you have the hypothalamic axons, they send out hypothalamic releasing hormones into the anterior pituitary.
No, into the
Median
median eminence.
eminence, and then like it stimulates anterior pituitary.
Right, because of those releasing hormones are carried by the portal circulation to the anterior pituitary.
Yes. And so, but for the posterior part, we say that like vasopressin and oxytocin are posterior pituitary hormones.
That's right.
But in the initial lecture, we were saying how it was like this similar story to the anterior pituitary. So is it the posterior pituitary that is making these hormones, or is it the hypothalamic axons that go through the median eminence and reach the posterior and then release from the posterior pituitary?
It's really the latter. So the posterior pituitary is really an outcrop of the hypothalamus.
Okay, yeah.
The cell bodies here, the cell bodies of the axons that release oxytocin and vasopressin are here.
Okay.
The cell bodies of the cells, neuroendocrine cells, that release posterior pituitary hormones, vasopressin and oxytocin, are here. This is where the cell body is.
The hypothalamic neurons.
Yeah, the hypothalamic neurons project to the posterior pituitary where they release their hormones. So the posterior pituitary does not have vasopressin, cells that release vasopressin, or cells that release oxytocin. They have terminals of hypothalamic neurons that release those hormones.
Okay. So technically, the posterior is kind of the release site.
Of some hypothalamic hormones.
Yeah.
The other release site is in the median eminence of the hormones that trigger the cells of the anterior pituitary to release their hormones. Here's another slide from early in this section that sort of flow charts. This is another, this is from the textbook that flow charts the hypothalamic hormones that are released into the median eminence, that travel to the anterior pituitary and trigger specific cells, gonadotropes, somatotrophs, thyrotropes, lactotropes, corticotropes, to release their hormones, FSH and LH, growth hormone, TSH, prolactin, and ACTH. There are 1, 2, 3, 4 of these are positive feed forward, and 2 of them are negative feed forward. And then the effects of LH and FSH in the body, growth hormone in the body, TSH in the body, prolactin in the body, and ACTH in the body. Question?
When it comes to the positive and negative feed forward and feedback, are you going to want us to know which ones do feed forward and which ones feed back?
Yeah.
Okay.
So I am going to ask you to know specifics of feed forward and feedback. This is general. And this is slightly modified, I think, from the slide that I showed you earlier, because it shows that in the positive feed forward, sometimes the effector is downstream of a gland, right? So the effector here would be, let's just talk about thyroid. TRH stimulates the thyrotrophs to release TSH, stimulates the thyroid to release T3 and T4, has pleiotropic effects on developing neural cells, muscle, heat-generating cells, fat, lots of effects. I do want you to know some of the effects so that you can understand, okay, if someone's hyperthyroid, they're going to be running hot, and they're going to be maybe having fast heart rate. I don't expect you to know all the details of hyper and hypo thyroid or hypo TSH or SH or whatever, but I do expect you to be able to solve some problems with classic symptoms like running hot, running cold, too much cortisol, too low cortisol. Did that answer your question? Yeah, wasn't that your question?
Well, actually, I think what I'm most wondering is it always gonna have the long loop feedback and the short loop feedback? Or some of them maybe feed forward?
I guess they're probably, they all have feed forward. Sometimes there's a negative feed forward, like dopamine controls the lactotrophs of the anterior pituitary and limits the release of prolactin, which limits the secretion of milk until that negative feed forward inhibition is relieved. Specifically, I don't know for sure whether there is short loop feedback of prolactin or long loop feedback of milk. So no, we don't always have long loop and short loop, but unless it's obvious that you don't. There's no way that. So let's see. Dopamine, prolactin. I think prolactin does have short loop feedback, but and you could just sort of say, okay, let me assume that prolactin has short loop feedback because that is so common. Prolactin. The other gland here is the mammary gland, releasing milk. Well, there's no way that milk has negative feedback, but there could be other things that are released by the mammary glands. So, I guess when in doubt, you should assume there's both long loop and short loop negative feedback. You'll be able to answer a question correctly if you make that assumption. We can do some clickers if you want, just to get a conversation going. Yes?
Is there going to be any anatomy other than the anatomy of the endocrine system? Or
Not
just
on this exam. The anatomy on this exam, so the anatomy in the quiz is obviously the anatomy on the list. The anatomy in this exam will focus on endocrine and male and female reproductive anatomy. A lot of correct answers, indeed. Any questions? Which of the following hormones do not follow a feed-forward pathway? Do not follow a specific feed-forward pathway. Hypothalamus, pituitary, gland. which of these pathways do not have a hypothalamic releasing hormone and a pituitary hormone and a other gland hormone? Any questions about this one? This one is harder to parse. Cortisol does, right? CRH, ACTH, cortisol from the adrenal. Thyroid does. TRH, TSH, T3 and T4 from the thyroid. Insulin does not. Insulin, the cells of the beta cells of the pancreas detect glucose and in high glucose release insulin. There's no hypothalamic or pituitary component to that feed forward. It's really not even feed forward. Detects glucose and releases insulin. Parathyroid, the same thing. Detects calcium and releases parathyroid. Detects low calcium. Okay, we can do a case study. I mean, like I said, this is not to cover material as much as it is to stimulate learning and conversation. So we can do a case study. They're interesting. I'll read it out loud. Well, this is just background. If needed on. So those are the results. I haven't looked at this in a year. I must be on this as myself. Let's take the, yes.
What's the difference between total and free
What, okay, this would not be on the test, but total is the amount of T4 that is in the serum that includes both unbound and T4 that is bound to a carrying protein. So many hormones have both carrier proteins and are freely soluble. And so free T4 is the shorthand for unbound T4. T4 that is free in solution. Bound T4 would be T4 that is bound to a carrier protein. And total is free plus bound. So let me just look at the next slide. So I think what we're supposed to do here is just talk about the options. Which of them are plausible? It's not a clicker, I don't think. So let's talk about them. Let's start. Does it make sense that is she hyperthyroid? Is she showing signs of hyperthyroid? Yeah, well, I mean, right here, by definition, this is the sign of hyperthyroid. Why is TSH decreased? Why is TSH decreased? Negative feedback. So, could there be, so is A plausible? Given these symptoms and these signs, is A likely?
I'm just asking a question.
Yeah.
If there's negative feedback from T4 on TSH, would TSH then have, like, allow TRH to build up, or would T4 also negatively feedback?
T4 negatively feedbacks onto the, there is both of them. Huh?
I didn't even answer the question, I was just asking.
Okay. Let's, somebody then volunteer to answer the question. Is A likely? I'm not saying it's impossible, but is that a likelihood? No, it's not likely. Why? Negative feedback. I mean, the question is, do you expect TRH to be high? Well, we have reason to think that it isn't. I mean, we would have high negative feedback, and we have low TSH. Low TSH. Low TSH means it's, I mean, negative feedback hardly ever overrides positive feed forward. So a hypothalamic tumor is going to produce boatloads of TRH, which would then lead to increased TSH, not decreased TSH. You could argue around it. You could say negative feedback is overriding the positive feed forward, and that's just not likely. So A is not very likely. Increased secretion of TSH from an anterior pituitary hormone tumor. Is that likely? No. TSH is decreased. So B is not very likely. Primary hyperactivity, primary hyperthyroid, Graves' disease. Is that plausible? Yeah. Ingestion of exogenous thyroid hormones. This is, people do this for weight loss. Is that plausible? Can't rule it out. The physician didn't ask her. Your model might be motivated to keep weight loss down, and maybe she's taking too much of a thyroid supplement obtained through less than medical routes. So let's see what the next slide is. Okay, C is most likely, but we can't rule out D. We just can't. Here, I mean, this is a word I didn't use in class, thyrotoxicosis. Shall we unpack that one? Thyroid, toxic levels of thyroid in the blood. Osis. Emia really means in the blood. Osis just means that's what we're talking about. Questions about this, the logic to this problem? Okay, it continues. I forgot about this part too. Natasha's thyroidectomy. What is that? Removal of the thyroid. Was successful and she's recovering well. Her nervousness and palpitations have disappeared. She's gaining weight. Her blood pressure has returned to normal. She begins to experience alarming new symptoms, however, including muscle cramps, tingling of fingers and toes, numbness around her mouth. I didn't make this up. This has got to be a real study. She returned to her endocrinologist and found that her total blood calcium was 7.8 milligrams per deciliter, and her ionized calcium, so this is total and free. In the case of calcium, we talk about total and ionized. The total including calcium that is bound up in other things, and ionized being free, dissolved calcium. A total calcium of 7.8 mg per deciliter and ionized calcium was 3.8 mg per deciliter, both of which are lower than normal. Hypocalcemia are 9 and 5. Okay, what may be causing hypocalcemia? Loss of the parathyroid, right? You take out the thyroid gland, you take out the parathyroid. What should be done? Dietary supplements with parathyroid hormone, which are possible just like there are with thyroid hormone, I think.
Can it be, calcium supplements directly?
Calcium supplements directly might help, but not enough. You still need parathyroid hormone to maintain homeostasis. So this is a setting not of negative feedback, but of antagonistic control, right? So here is a setting. Here is a cartoon of normal antagonistic control of calcium with a set point of 5 mg per deciliter. This would be the ionized calcium. And when we remove parathyroid hormone by removing parathyroid cells, we now have less power, less possibility to generate parathyroid hormone, and so the set point moves to a lower set point, and we have hypocalcemia. Yes?
Isn't that also affect calcitonin too, then, because the source of it is the
Yeah, so everything would be, so that's true. Maybe this case study, I'm trying to think of why. Yeah, so the calcitonin would also be lower, so that might shift this set point further up than this cartoon is showing. But apparently, in this, the removal of both parathyroid and calcitonin, the loss of parathyroid is more important than the loss of calcitonin, and you get hypocalcemia. I'm not expecting you to fully grasp why low blood calcium would explain muscle cramps, tingling, numbness, if that's complicated. So that's not a set of symptoms that I would expect you to get. But I do expect you to be able to remember concepts of antagonistic control. Insulin glucagon, parathyroid hormone, calcitonin, somatostatin, GHRH. Yes, Al?
Are calcium from like the bones or wherever getting it from?
Yeah, it's getting it from the bones. Questions? Shall I just keep moving forward through what I got? Okay, some more clickers. This is an interesting problem. Which of the following drug types could be effective in treating insulin-sensitive diabetes mellitus? Insulin sensitive, this is type 1 diabetes, this is because of loss of the pancreatic beta cells. Insulin sensitive means this is a person whose blood glucose is normalized when they receive insulin injections. You may have heard that there's big news in the world today about the cost of insulin just going crazy. That's a symptom of a broken healthcare system. Okay, which of these could be helpful in a setting where there is low insulin release? Now, this is not complete type 1 diabetes. This is low insulin release. There are still, let me just set this up. There are still beta cells in this person's pancreas. let's go through them. A, is A a possibility? The blocking calcium channels increase insulin release. Hard to see how. A drug that inhibits the GLUT2 transporter. Well, I'd have to show you a model, but remember I'll just tell you, the GLUT2 transporter is the uniporter that brings glucose into the pancreatic beta cell. So is the drug that inhibits that going to help the beta cell release insulin? Hard to see how. A drug that inhibits leak sodium channels. That could do it, right? Because inhibiting a leak sodium channel, no. A drug that inhibits the leak sodium channel is responsible for depolarization. Right? I mean, resting potassium channels and ATP-sensitive potassium channels are responsible for maintaining a negative resting memory potential, and a leak sodium channel is responsible for not having as negative a resting memory potential, a somewhat depolarized resting memory potential. If you didn't have leak channels, the membrane potential, leak sodium channels, the membrane potential would be more negative. So blocking leak sodium channels is going to make the resting membrane potential more negative. Hard to see how that's going to help insulin release. A drug that blocks the ATP blocked potassium channel, is that going to be helpful? Yeah, that's the one. Why is that one? The ATP-sensitive potassium channel is a contributor to the negative resting membrane potential of pancreatic beta cells. ATP, produced by oxidative phosphorylation and other pathways of respiration, stimulated by glucose, blocks that channel. ATP blocked. It's going to allow the cell to be more positive. More positive means depolarization. Depolarization means calcium channels are activated. Insulin release. So a drug, and there are drugs on the market now, I forget the name of them, but there's a bunch of drugs on the market that augment the ability of the pancreatic beta cells to release insulin by blocking the channel that is blocked by ATP. Which of the following drugs could enhance glucose uptake? Just as a reminder, GLUT4 is the glucose transporter in liver and muscle that allows those cells to take up glucose. So I sort of gave you one answer right there. Let's see what else. I haven't looked at these in a year. I think right now, I would have selected all four. Let's go through. A drug that allows the cells that take up glucose to take up more glucose. A drug that activates the receptor that allows the cells to take up more glucose. It's pretty obvious. This receptor activates this pathway, so a drug that just bypasses the receptor and activates the pathway would allow the cell to take up more glucose. And a drug that slows the turning off, slows tyrosine D phosphorylation, slows the turning off of the pathway or of the receptors, both of which are tyrosine kinases. A drug that slows the turn off, augments the turn on, and that would enhance glucose uptake. So I think I have some slides. So here's a slide that reminds you of this is the pathway that this question is getting at. let's look at it. Any questions about that one? And this is the pathway that the second question is getting at. Insulin receptor, MAP kinase, tyrosine phosphorylation, gene expression, including increased expression of GLUT4, more glucose intake, more glycogen and fat production.
Oh, can you go back to the first question?
So that's a drug that, let's take them apart one at a time with the cartoon up. A drug that inhibits activation of voltage-gated calcium channels. Well, if we block voltage-gated calcium channels, we're gonna block hormone release, that's pretty easy. A drug that inhibits the GLUT2 transporter, a drug that blocks the glucose uptake, is not gonna allow the cell to make the ATP, is not gonna block, da-da-da. Oops. A drug that inhibits sodium leak channels, well, that's, I mean, there's no such thing. But remember, it's now, I'm asking you to remember that it's sodium leak that allows inhibition of a potassium channel to cause depolarization. If we don't have sodium leaks, this doesn't happen.
I just wanted to look more into the ATP synthesis,
Okay.
blocking, I'm thinking of that.
Okay?
So blocking the ATP sensitive potassium channel, so this channel brings in potassium.
This channel lets potassium
So
out.
out,
That's
okay.
always true.
And so it's blocking the whole, so it's not blocking the, I just don't get the point of the drug if we have GLUT2 coming in and we have
Yeah.
ATP production, normal ATP production, the channel would be blocked
Well,
with
I mean,
ATP.
oh, yeah, but the channel's not always blocked. The level of ATP is not, so this is a dose response thing. The level of ATP is not enough to block all this channel. Some of it is blocked, that usually, typically, normally causes enough depolarization to release enough insulin. In this patient, that amount of block is insufficient to cause enough insulin release. So we are going to augment the block by ATP by providing another thing that blocks it, the drug.
So we can assume that the deficit is in the glucose, or, I mean, it could be in the glucose uptake or in, oxidative phosphorylation for ATP production.
Yeah, it could be.
Okay.
So, but it's simply bypassing what could be upstream. And it could be a lack of the K-channel response to ATP. This could be normal, this could be normal, this could just be not responding to ATP normally. And so we're going to block it with a drug instead of with ATP. Yes?
What were you saying about sodium leak?
So, remember this is really review for the final exam. Arresting membrane potential is a balance between potassium efflux through whatever potassium channels there are and sodium influx between whatever sodium permeabilities there are. That's the balance that leads to a resting membrane potential. If we block potassium efflux, then the effect of sodium influx is greater and we have depolarization. If we block sodium influx, then the effect of potassium efflux is greater and we would have hyperpolarization. So a drug that blocks leak sodium channels would not be effective because it would cause hyperpolarization, not depolarization. A drug that activates, if we could activate leak sodium channels, that could be effective. That would bypass all of this and lead to augmented depolarization. But those things don't exist. This is a thought experiment, not a real experiment. Okay. Well, that's all I got. plenty of time for more questions.
You said we didn't have to know about inhibition in the,
That's right. I'm not gonna ask you a question about it.
so therefore, like, a concept about, like, FSH eventually, like, you wouldn't ask us about, like, FSH eventually leading to, like, negative feedback of gonadotropes? Is that something
I
that we
would-
should just know without knowing the word inhibin?
Yeah.
Okay.
I do want you to know about negative feedback. I don't carry that you memorize the word. So, just to summarize, kind of, keep the conversation going, the slides that I've put up here have focused on the physiology that I want you to know. The thalamic, pituitary, other hormone, the HP axis, I put down, not just HPA, but the other hypothalamic, pituitary, other effector story. It's a major part of what we talked about. That's what I just said. I do want you to be able to solve problems using that knowledge. I would not give you a case study, at least not one that took this long to read, because this is not a reading test. But I might give you a shorter case study and ask you to solve a problem with it. I could ask you to be able to interpret a graph like this, and why loss of one hormone or the other could lead to hyper or hypo levels of the controlled point. Yeah.
Should we notice that code for these things?
Oh, no. You mean the actual numbers?
Oh, no, what is ultra short feedback?
Did I put that on one of my
Yeah.
notes?
It's
Ultra
like-
short feedback is when hypothalamic releasing hormones inhibit their own release.
Okay. Do we know if any of them that think they all do that, or?
I'm surprised I left that in there. I don't know about that anymore. Yes, can I explain that?
Yeah, we explain it on
Okay. Try to walk through why these hormones lead to a set point? By,
I know parathyroid increases.
that's right.
Yeah.
Parathyroid hormone causes the increase of blood.
So you think it like go up, like the longer you go up.
Well, so this is not the calcium concentration. This is the calcium, so this can be confusing. This is the calcium concentration. So this is the independent variable, and what's plotted as the dependent variable is the level of the hormone. So at low serum calcium, the level of the hormone, of parathyroid hormone, is high, and it causes calcium to rise. So we slide along this axis. Low serum calcium causes parathyroid hormone to be high, which causes calcium to be released from bone and increases calcium reabsorption in the kidney and in the intestine. So serum calcium concentrations go up. Low serum calcium causes a low concentration of calcitonin. And so calcitonin's effect is low, Parathyroid's effect is high and calcium concentration goes up. Let's go to the other extreme. At high serum calcium, that causes low parathyroid hormone concentration because it inhibits parathyroid hormone from being released, and it stimulates the parafollicular cells of the thyroid to release calcitonin, which causes decreased release of calcium and increased excretion of calcium. So calcium concentration in the body goes down. So at a set point where these antagonistic effects are balanced, we're at a stable, it's not an equilibrium, it's a stable set point. There's a word for it that's better than that. I'm blanking on it. Steady state. Stable, steady state. And we might oscillate. We might go a little low, which causes calcium. If the calcium concentration were to go low, then parathyroid hormone concentration would be a little higher. The effect would be a little higher than calcitonins, which would cause calcium concentration to go up. And then we might overshoot. And then that would cause the calcitonin effect to be greater than the parathyroid hormone effect to be. And that would cause calcium concentration to go down, and we might overshoot. So there could be some oscillation around the set point. But what we're talking about is a stable, steady state. Is that helpful? These were just to remind you that we're not only talking about hypothalamic, pituitary, other glands stories. There are some other stories involved with detection of the thing, glucose, calcium, directly, not by the hypothalamus, but by the endocrine cells themselves. So this was all physiology, but let me just make it worth your while to remember that anatomy will be on the exam, anatomy of endocrine as well as male and female reproductive endocrinology. I mean, anatomy.
By endocrine, do you mean like where it's released from, or what do you mean by the anatomy of endocrine?
Pardon?
Like by anatomy of endocrine, do you mean like where it's released from, or do you
Yeah.
really know like this is what it looks like, or what would be an example of that?
Yeah, so, I mean, how could I ask you a question that was related to endocrinology, anatomy? I mean, I could show this picture with the boxes filled in or blanked out and say, okay, name a hormone or two or three that's released here. Name this structure. I could see asking you to label this image. Or I could ask you to label, I mean, I could pull up this, the picture of the male or female reproductive organs and then ask you to label those, or the male and female reproductive cell, the cell types in the testes or the cell types in the ovary, and ask you to label those. Those could be on the exam.
Things like the thyroid or the cortex.
What could I do with the thyroid? I mean, I could show a picture of the body and say, point to where the thyroid gland is, and ask you that. I mean, that just seems kind of Mickey Mouse. Yeah.
Can you re-explain the median eminence in the infundibulum? 'Cause
Yeah, okay.
Last time you said that the structure is called the infundibulum. Yeah.
It isn't a point worth worrying about.
Okay.
The name of this stalk. So one name is a anatomical name. The name of the stalk, the thing that is broken in the cadaver brain, is the infundibulum. The median eminence are the secretion sites in the infundibulum. So this is a physiological name. Infundibulum is a purely anatomical name. That's the difference. I want to be mindful of your time. You you have other things to do or things to do for this class, I'm happy to just sort of close up shop here. I'll be in my office, and so if you have questions that you'd like to ask me, one-on-one, I'll be available for the next, well, for quite a lot of the day, actually. There is the cadaver open house that I'm gonna open up as soon as we're done in here. And then I'll shut down the cadaver open house late in the afternoon, because I have to get ready for this evening's practical. Remember that some people will be taking the practical today, and some people will be taking tonight, and some people will be taking the practical next week. If you're, I just ask you, and you, people at home, to not put your friends in the difficult position of asking them questions. Don't tell your friends. This is a don't ask, don't tell situation. Don't ask your friends anything about the practical. Don't make it uncomfortable for them. And if you've taken it, don't tell your friends. Don't violate the process.